right, so today we are talking with Dr. Ryan Tam, a graduate student at North Carolina State University in the Chemical and Biomolecular Engineering Department. He recently published a paper entitled Human Pluripotent Stem Cell-Derived Medium Spiny Neuron-like Cells Exhibit Gene Desensitization. This was published in Cells in April of 2022. So thank you so much for talking with us today. Just to get started and learn a little bit about you and your background, how did you get in the field of stem cell research, and what made you decide to get a Ph.D. and continue on with research?
It's been a pretty circuitous journey, but I was originally a nano engineer at UC San Diego, and there was no biology involved at that point. It was more materials science / physics focused, but decided to get a minor in biochemistry in undergrad and really enjoyed that, but realized that a lot of the undergrad skills and knowledge I learned didn't really transfer, so I needed to get some experience. Worked at a small biotech company for a year, got a hang of things, and went to graduate school since I realized I needed greater qualifications to be able to do more of the things that I wanted to. So I looked around specifically for synthetic biology related labs, and NC State fit the bill in addition to being within chemical engineering department. Dr. Kyung does, or previously did, because I joined his lab only a year after he started, did some previous stem cell engineering work, and he had just started up this portion of his lab. So it seemed like a really good opportunity to work on stem cell models, synthetic biology, that is applicable to the discovery, potential discovery at least, of things that may help address human diseases. In this case, it was a grant that he had received for doing basic research on addiction. And so that's how I ended up at Dr. Kyung's lab.
Yeah, so on the subject of stem cells specifically, do you guys get pushback using stem cells as a model for like substance use disorder or anything, or just using stem cells in general, or have you guys found it pretty easy to be in that field?
It's pretty easy in the position of a graduate student, and as far as I can tell, even as from Dr. Kyung's perspective, he hasn't had any pushback, considering how ubiquitous it is to use some of these stem cells. As well as our work isn't really that public facing, so it's not something that people would immediately latch onto, since we're just using stem cell stocks that other companies have made and built up, and we don't get them directly from patients, for example. So no.
So do you partner with these companies, or is it more of like they sell them and you just buy them? Okay.
The latter, they sell them.
Okay. So you are towards the end of your graduate student career. Do you have advice for someone who's either, thinking about going to graduate school and doesn't know if it's the right choice, or is early on in their graduate school career?
Yeah, I'd say the biggest advice would be that you have a solid reason for going to graduate school. Going in to graduate school, imagining that it'll give you all the answers along the way is probably not the way to do it. You want to go in knowing full well why you're going in, so that you don't regret it somewhere in the middle. I've seen examples of this over my graduate student career. And it just becomes a race to get to the end rather than to try and do impactful science for whatever ultimate purpose you went into graduate school for. Another small tidbit of advice is that even if you don't end up working in your dream lab in the department you sign up for, everything's okay, since it's more about the skills and the process of learning how to learn in graduate school that's most important, not specifically what you work on. That can be, you can learn new things in your new positions as long as you can exhibit the important ability to think on your feet and know how to obtain information relevant to the scientific question at hand.
Yeah, that I do see a lot of people getting stuck in the minutia, but yeah, looking at the big picture, that's good advice. So moving into your paper a little bit, can you describe the major findings of this paper for anyone who's watching and maybe hasn't read it?
Yeah. So this paper focused on using stem cells and turning them into neurons of a specific type that are implicated in the development of addictive behaviors. There hasn't been any prior work observing characteristics of these types of cells, so we sought to investigate them, and they are of interest because they are human as opposed to rodent, which a lot of the addiction work is done in rodent models. So we wanted to pair up a lot of that rodent data with potentially relevant human-specific mechanisms to similar perturbations that would occur in the brain in response to drugs of abuse. So specifically, when drugs of abuse are introduced into the brain, these cell types, medium spiny neurons, are exposed to these drugs, and ultimately the amount, it is hypothesized at least, the amount of dopamine that is exposed onto these medium spiny neurons is increased as a direct result or indirect result of drugs of abuse. So we wanted to investigate how these human systems responded to dopamine as sort of a proxy to drugs of abuse, given our model system. It's in vitro, it's not on an organism level or even a whole brain specific level. So we needed to come up with the conditions that best allowed us to obtain interesting and useful results within this model system. And so what we found was that these stem cell models can in fact respond to dopamine in a way that has been predicted previously in rodent models. Of course, the overlap is not 100%, potentially due to the differences in the model or potentially due to species-specific differences. And we see that we can recapitulate certain phenotypes. One important phenotype that has been talked about in addiction literature in specific relation to molecular phenotypes is a phenomenon known as gene desensitization, where upon the introduction or perturbation of these cells to a single acute dose to drugs of abuse such as cocaine, there is a certain level of gene expression, but this level of gene expression reduces over a repeated dosing regimen of drugs of abuse. In our case, since we're using dopamine as a proxy, we show that there seems to be some level of gene desensitization after a chronic dopamine dosage as compared to an acute dopamine dosage. And we show that the genes that are shown to be desensitized tend to overlap, at least functionally, with what has been shown or observed in rodent models. We also took a step further to further evaluate subtype specific or more complex responses in these medium spiny neuron models by looking at their response to not only dopamine, but dopamine's agonist, such as a D2 agonist, and the complex interplay that 2D2 agonist receptor may have with another agonist that targets a receptor that shows complex interplay with these dopamine receptors. And so we wanted to demonstrate not only this desensitization phenotype, but also this more complex intracellular interaction. We also did some further optimization with what we define as a chronic dosage, since a lot of the rodent work defines chronic dosage in a certain way, but similar definition has not been established in these stem cell models. So overall, if I had to give a sort of TLDR, this paper has to do with characterizing responses to dopamine because they are a relevant perturbation that these cell types see in the brain, and with that, these cell types do seem to recapitulate certain phenotypes of responses to drugs of abuse, and that this is the first example of characterizing this type of model in this way.
So is gene desensitization that's obviously, as you described, seen in rodent models in a similar way as what you showed. Is this correlated with symptom or symptomology in any way? So and would drugging that or targeting it with some sort of drug change the symptomology, or is this understanding the disorder mechanistically? Is that more what this is focused on?
Yeah, the focus is more understanding the mechanisms that underlie this gene desensitization. But the first step with these models is to show that we can, in fact, see this phenotype. And then later on, further work will focus more on how can we reverse this phenotype, and would it also affect other phenotypes associated with gene desensitization or chronic dopamine dosage. So, for example, in this paper, we only focus on transcriptional phenotypes, but there are, of course, other ways that we can analyze the response of these cells to dopamines, such as functional phenotypes like calcium imaging or electrophysiology, or morphological differences due to dopamine. So I think that is a future aspect of this work where we want to set up the basis for which we can then interrogate and maybe reverse gene desensitization in later work.
Yeah, so you talk a lot about like the role of dopamine in this system, and in the paper you discussed that it's not only critical for substance use disorder, but it's also we also see perturbations in like deficits of learning and memory. Can you describe like why dopamine, or how dopamine is so central in these models?
Yeah, specifically within this model, this medium spiny neuron model, it has to do with the neurotransmitters that it primarily responds to. So the neurotransmitters that these neurons respond to include dopamine, glutamate, acetylcholine as the primary ones. But there has been a lot of research shown that dopamine serves an important purpose to drive some of the behavioral phenotypes that are altered in diseases such as Parkinson's disease or addiction. So there is a large focus in the field on dopamine specifically as a neurotransmitter that causes changes in intracellular signaling pathways that ultimately leads to these changes, but it is more understood on a rodent circuit specific level, and few investigations have been performed both in human systems and in these particular cell types. So the emphasis is on dopamine because of a lot of the previous research, but the question in these human systems is whether we can see a similar impact. And so we wanted to investigate dopamine first, and then later papers investigate the effect of other neurotransmitters on a molecular level, on a cellular level, to see if similar findings could be extrapolated in a human system.
Yeah, that makes sense. So one thing that a lot of people get pushback on in like modeling with rodents and using ex vivo tissue for rodents and that type of stuff is that it can't model the complexity of like a where there's all these different glia and neurons. And obviously stem cells has more complexity because they can differentiate into multiple different cell types. How are you able to verify this model that models something that we can't really get a good idea of in something like a rodent model? Like, how can you verify that that's more close to a human brain system?
Right. Yeah, I mean, that's one of the main challenges of this type of work. It's an extension towards ultimately better modeling a human brain without fully getting there, because it's a limitation of these types of models. So we can only do as much as we can to validate that, for example, the cell types that we get in the end express the receptors or specific markers that should be expressed by these cell types.
Yeah.
And have sort of a understanding that these are obtained from a human system, and that we may be able to at least compare this model from what's been shown in literature and see what the differences are and evaluate whether it is a valid model. So it's more of a investigation into the validity of these types of models.
Yeah, so stem cells are less frequently used in modeling systems than rodent models, but they are obviously of critical importance. What role do you see stem cells being able to play in the modeling system that would pair well with all the other modeling systems we have currently?
I'd say for the work that I do, at least, there is some potential for gaining, first of all, getting a better understanding of these types of cells, and potentially being able to better understand human specific mechanisms in addiction or other diseases implicated in these cell types, uh, as a supplement to a lot of the work that's already being done in other models. So it wouldn't be a replacement of any other model. It's not what we're proposing. It's more of a to help us fill in the gaps in knowledge. So that, I believe, is one aspect we can use these types of stem cell models for. Another aspect would be potentially for stem cell type therapies. So in a distant future, of course, there might be some therapies that require the replenishment or reseeding of, say, these certain cell types. And if that were to be the case, then our work would help to establish a baseline for some of the characteristics for these cell types before their introduction into the brain. Of course, the context of introducing these stem cell models into the brain would be completely different, but we believe our work sets a path towards that type of usage.
Yeah, so you discuss a lot about how critical it is to translate the research from rodent to human. But what about, so you obviously agreed to do this interview today. How do you see like translating the research from, you know, an academic setting to like a public setting? Do you see that as critical as, you know, doing the actual on-hand research, or do you think that you know, the current state of it is sufficient for the understanding of the public?
Definitely, yeah, not sufficient. I think it plays both directions. We both have to gain a much better understanding of these model systems before we can actually be able to use them for the development of therapeutics. In addition to whatever the specifics of those therapeutics that might eventually be developed, figuring out you know, more on a more practical realm, figuring out the best way to manufacture them, cost-efficient way, and whether the therapeutics that would be developed from using these model systems, would they be different from traditional therapeutics, or would they be similar? That's an ultimate question that down the line, if development of these specific therapeutics extrapolated from work with medium spiny neuron models, would ultimately have to answer, but that's unfortunately not something that I have an answer or even an inclination to at the moment, considering the amount of time that I think these models need for further development.
Yeah, so a few, you know, rapid questions. So what are some exciting research areas in your field, or, you know, in the field in general, that you see happening on the horizon that you're like excited to come out or see develop?
Right, yeah. I think the primary thing in the addiction field, at least, is the focus on epigenetics of addiction. So there is the theory that certain epigenetic markers may predispose the development of certain molecular phenotypes and ultimately cellular and behavioral phenotypes. That there is a lot of work being done, say, for example, in the Eric Nesser lab, to better understand some of these epigenetic phenomena. So I think that's a very exciting direction that might potentially lead to therapeutics that are ultimately more effective for, say, the treatment of addictive behavior, specifically to better understanding why certain individuals are more inclined to be addicted to drugs of abuse and why others aren't. And there is a hypothesis that epigenetics may be involved, so that those developments are very exciting. In the stem cell field, the development of and investigation of more of these models, not only on a 2D format, but also in 3D models such as organoids, and further development of organoid models and advancement of their complexity, reduction of the homogeneity or the heterogeneity of these organoids, a lot of this research is exciting because it presents the opportunity for generating these new models that may have ultimately much better insights into, say, the human brain than previous models have. So a lot of this work, both in the addiction field and the stem cell field, seems exciting to me, at least.
So you're obviously at the tail end of your graduate student career. What comes next for you?
Yeah, I'm gonna be working at a pharmaceutical company, a startup here out in Durham, North Carolina, Tune Therapeutics, which focuses on developing epigenetic therapies. So it'll be somewhat related to the epigenetic work that I had done, or a little bit of what I've learned in graduate school, and a lot of the same molecular assays and skills that I had obtained over the course of my graduate student career would be applied to this position. So I'm excited for this next stage in my career.
Yeah, well, congratulations on that, and thank you so much for talking with us today.
Yes, thank you so much, Amara.